This tutorial will give you just enough information to run an Anova analysis for Bio 1 or Bio 2. An Anova analysis often includes categorical data on one axis and numerical data on the other. However, in Bio 1, we may end up using it for some of our scatter plot data that has numerical data on both axes. The first thing we do is we input our data, and I downloaded the Anova template from the lab website, and I pasted it here, and we can see there's 3 tabs: Anova, Graph, and Tukey-Kramer. This is the page I would do my graph on. I am not going to do the graph, I'm just going to analyze the data for now. So the first thing we do is we copy this table. So we highlight it and we copy it. Now we need to paste this table, but we need to change its orientation to run the analysis. So to do that, I click on Home up here and I go to Paste and Paste Special. I then go down and click Transpose. Now notice what we've done. On this original table, we have water, fertilizer, bleach, and soda are independent variables running vertically here. Now they're horizontally across the page. And we've also flipped the orientation of the replications. They were horizontal, now they're vertical. So we flip the table around. So now what I do is I highlight my treatments and I copy them: water, fertilizer, bleach, and soda. I go down here to the Anova tab, and I'm going to paste them right here where it says enter your group names. Those are the treatments. So we've grown plants using different treatments. Water is the control. We use fertilizer, bleach, and soda. So maybe Coca-Cola or something. We've watered our plants. We're looking for growth over a week-long period, and that's what we've measured. So I'm going to highlight that data here, and I'm going to paste it in the same table here. All right, we're almost done. Scroll up and look right here where it says P. This p-value is an important number. If that number is greater than 0.05, that would indicate that none of your treatments had any significant impact. They were all the same. They all had the same amount of growth, statistically speaking. So what we're doing here is we're trying to figure out which types of treatments had significant impacts on growth, because in science you only use the word significant if you can back it up with statistics. It's very important you don't use that word just whenever you feel like it. It has a very direct and distinct meaning in science. So we know here that the value, this p-value, is much less than 0.05. So that means at least one of our treatments had a significant impact on growth. So to figure out which one, we go to this next tab called Tukey-Kramer. This is a post-hoc analysis, meaning an analysis of the data from the Anova. And we're going to then be able to use it to figure out which treatments were different. Let me zoom in on this real quick. Now what you need to do is only look at these values that I'm highlighting in yellow. I'm just highlighting these to make it clear which ones we need to focus on. Now, have a look at this data. Let me scroll over. Starting down here with fertilizer, we can't compare water to water. We can't compare water to water, so it's just blank right there. And these numbers are not used in this analysis. So starting here with fertilizer, this is the fertilizer row, and this is the water column. Scroll over to water. We're not even going to pay attention to the number. You can learn about that in stats later on, but there's an asterisk right there next to the value. That asterisk indicates that water and fertilizer have significantly different amounts of growth. So you can use the word significant. So that's how you figure that out. So bleach in this case, significantly different from water. Bleach also significantly different from fertilizer. Soda, significantly different from water, significantly different from fertilizer, but not significantly different from bleach. See how there's no asterisk there. So only pay attention to the values that are below these dashed lines, right? So look at this, these dashed lines. These are the values that we use right here. And you could then comment on which ones were significantly different in your written results section of a lab report, or you may make some comment about it in your figure caption as well. Although I wouldn't talk about every single treatment in your caption, you could highlight anything important that you wish to discuss about the trends and the important significant differences in your data in that caption. You will also add in error bars, but I just wanted to point out that we actually use the results of these tests, like a t-test or ANOVA or maybe a regression, to determine significance. We don't use that word lightly in biology. We tend to not even use it when looking at error bars, because error bars give us an idea if it's likely if something is significant or not. If the error bars overlap, it's likely not significant. If they don't overlap, it's likely they are significantly different. But we really need to hammer it home, and the only way that you can really use that word significance is by running a test like this. All right, I hope you can follow that, and we'll try this out in class.